Finally, we're at the Senior Investigator Award winner, which is presented to an established researcher for outstanding continued contributions to the field. Dr. Peter Yurchenco, this year's awardee, received his MD and PhD from Albert Einstein College of Medicine in the Bronx in 1976. He began his studies of basement membranes as a postdoctoral fellow in 1981 and has made it his life's work as a scientist. He joined the faculty at Robert Wood Johnson Med School at Rutgers University in 1984, where he has spent his entire 41-year independent career. Peter's made many seminal discoveries, but I'll just mention that although some were in the biochemistry of collagen, I think most of us know Peter as a laminin biochemist. I've known Peter since attending my first Basement Membranes Board conference in 1994, and I felt I was meeting a legend then. Yet he was only 10 years into his faculty position. 30 years later, I'm thrilled to be able to present this award to him. Please join me in congratulating Peter Yurchenco.
Thank you. Let me just say that this is a tremendous honor. I thank the Society and hold this dear to my heart. What I'm going to talk about spans many years, and so I'll have to make references to some earlier studies, even though I won't show the data, because I am trying to compress 42 years into 25 minutes, or is it 20 minutes? All right. No? That one. There's more than one green object on this. All right, so what I want to start with is a general model of basement membrane assembly. This reflects our work, but it also reflects the work of other investigators, some of whom are probably in this room tonight. It represents an effort to integrate in vitro work with genetic studies, mainly conducted in mice. And the model is basically as follows. A laminin is required to initiate basement membrane assembly, and it does so by binding to receptors on the surface of the cell, principally or almost entirely integrins and dystroglycan for two of the laminins. It also has attachments or potential attachments through sulfated glycolipids if they're present on the cell, but those are only present in the outer leaflet, so I think of them more as a glue to help assembly. Upon binding, this increases the local concentration on the surface of the cell and enables or promotes laminin polymerization, which is mediated through the short arms. This leads to the formation of a planar polymer that has stiffness to it. And we now know that signaling is greatly dependent on the formation of resistance to cytoskeletal elements provided by the polymer. Laminin also then recruits, that is, it has specific binding to other components, nitrogens 1 and 2, the heparin sulfate proteoglycan perlecan, and the heparin sulfate proteoglycan agrin. The nitrogen enables, but is the binding of type 4 collagen. Type 4 collagen forms a second polymer, and it does so through N-terminal and C-terminal interactions. You've heard earlier talks about this today. For example, Billy Hudson's discovery that the NC1 domain interaction requires the formation of sulfilamine bonds. In addition, early on, and this is really how I started my career, we found evidence for what I call lateral associations. There's supercoiling of the collagen triple helices that you see in tissues, you can also see in vitro. And if you think about it, even though it's likely a weak association, it's not covalent, but once you close off the N and C termini, they're locked into place, and they have the effect of increasing the tightness of the mesh. So now I want to turn to laminin polymerization. I discovered it and first reported it in 1985. And it's the result actually of an accident of incubating laminin at too high a concentration for what I was trying to get at. And what I observed is that it forms a flocculent precipitate. And when I examined the time course of this and concentration dependency, it became evident that it was a so-called nucleation propagation type of polymerization, in which there was an initial nucleation step of low affinity, followed by a propagation step that was calcium dependent and led to the formation of a planar polymer. Subsequent experiments using laminin fragments that contained the N-terminal LN domains revealed that the interactions were mediated solely by the LN domains, and importantly, it required an alpha, a beta, and a gamma LN interaction to form what we later called the polymer node, the tessellating unit of the polymer. Whereas the LG domains enabled laminin polymerization to occur on a cell surface. Now, if you look at the model I've shown here, it's a three-arm interaction model, it actually shows something that is expected to be thermodynamically unfavorable. You don't get many trimolecular collisions. And we knew that back then. We just weren't certain about the nature of the intermediate, other than it was probably a dimer or dimer replicates. So the next important set of, I should mention, this model was challenged in 2004 in terms of its specificity, but we were able to confirm this with recombinant laminins that were produced by my colleague, Karen McKee. It took a while to work out those conditions, but we introduced multiple LN and LN to LEA deletions and also did sort of domain swaps, finding that you really needed all three. The next set of major discoveries actually did not come from my lab, but it rather came from the lab of Erhard Hohenester at the Imperial College in London. He had a series of 3 papers that came out in fairly quick succession. And the first one showed the crystal structure for the alpha 5 LN to LEA domain. And what it shows, what he found, is that there's about a 120-degree angle between the LN and LEA. The back side of this contains a glycan. And the front side, he identified a few residues in a sort of patch region that appeared to be involved in laminin polymerization. And then he followed that by a publication of the LN-LEA structure for beta-1 and gamma-1, which are highly homologous. And then one other important step, he found out that you could take these recombinant fragments and separate monomers from dimers from trimers by high-resolution gel filtration. And I have to confess, at the time, I didn't think it would be possible to identify dimers that way because I thought the off rate would be too fast. Boy, was I wrong. So we picked up on this method. We increased the flow rate even further out to sort of the maximum of what you could get away with a column. And as you see here, yes, you could tell the trimers from the dimers from the monomers. And this allowed us to do incubations at different concentrations. We found a dissociation constant for the beta-gamma interaction, which was around 21 micromolar. And that interaction occurred in the presence of EDTA. That's our nucleation step. The trimer, though, required calcium. And calcium, as we had found earlier, and Erhard had localized to it was in the gamma-1 domain, but it's mediating not the beta-gamma interaction, but the gamma-alpha interaction that follows. So this led to, I think, a very useful realization. We could further identify the involved residues and determine which arms were interacting with which, because beta-gamma mutations for the bonding between those two should produce monomers only, regardless of whether or not the alpha subunit is present. But any mutations in either beta, gamma, and had to be an alpha should give us a dimer, not a monomer. All right, so one other thing. We started doing negative staining on these fragments and then doing image averaging, and that's shown in this slide. So in the upper part is the appearance of the monomers, and they kind of look like clubs. The dimer, they all look like monomers, and we're pretty sure what had happened, and it simply dissociated on us by the time we could actually visualize it by negative staining. The trimers, though, had this remarkable appearance of what I would say described as a near symmetrical triskelion. And if you look at one of the images, it actually, each subunit looks like a foot and an ankle at an angle. And it has toe to heel-like interactions, thinking of it as a heel. And we noticed that it would come in two versions on top of the mica. We didn't get side images. And so that suggested that this was a rather flat structure with this organization. Which heel bound to which toe? Well, we used that mutagenesis approach I told you. We picked conserved residues that were located on the face opposite the glycans. And this is gamma 1, and just showing examples. So a toe mutation produced monomers. That tells us the toe is binding to the beta chain, not to the alpha. Whereas mutations in the heel region, which are shown below, gave us dimers. Well, that tells us we're looking at an interaction with the alpha subunit. With beta mutations, everything was flipped. Toe mutations gave us dimer, so that means that's interacting with alpha, and heel mutations gave us monomers, telling us that that is the corresponding interaction with the gamma 1 toe. And as you would expect, both toe and heel mutations in the alpha subunit gave us dimers. So that gave us this crude map of interaction of which toe interacted with which heel. So the next thing we did is we collaborated with Arik Kulzik, who was located next door in the proteomics building, and recently had set up a lab for cryo-electron microscopy. Oh, and I should mention, Earhart had tried to crystallize the trimer, but he wasn't able to get crystals that diffracted. So we took this approach. And there was one other thing that helped us here. One of our mutations, it's gamma 1 D266R, actually didn't. We thought that would give us monomers, but instead it gave us a more stable dimer. And that's kind of shown in these gel filtration runs where if you ran it at a low flow rate, you would see the smear going across from the trimer position to the beta position, but that was prevented with this mutation. And it gave us the same triskelion structures by negative staining. So we ran with this because simply the time it took to pick up the fractions, measure the concentration, run across to the next building, and freeze down a pipette and freeze down the samples, it took at least half an hour, if not somewhat longer. Shown below are some of the earlier images where we were getting a better view of this triskelion. Working out which subunit was which, though, was a challenge because there's such tremendous backbone homology. But it was finally, Arik solved the structure. And what you see, he was able to identify now the interacting heel and toe contact points. They corresponded to nearly all of the residue mutations that we identified as being involved. The one exception was beta 1 F77, which, and to the right is shown the pi mole view from the front with those residues that were identified in mutations as being at the contact points. So in working out, okay, so there's another protein. It's called netrin-4. It's unlike netrin-1. It doesn't have the receptor interactions that netrin has, but it is a potent inhibitor of laminin polymerization. And this was first discovered in the lab of Manuel Koch, but I started collaborating with Raphael Reuten back in, about 2015, to look at the interactions with laminin. It has a C-terminal domain that can be eliminated and then it's just an LN and LEA or LE domain. It's very homologous to the other laminin LN to LEA domains. It inhibits polymerization, which is basement membrane assembly on cells. The interaction is much higher affinity than is occurring with the laminins. And to the right are the negative stain images I obtained, in which it kind of looks like a triskelion missing one of the subunits. It's got the same angle relationship. I used this interaction, knowing it was high affinity, to work out the conditions to then look at laminin. Well, Raphael showed that mutations in netrin-4 that are in the conserved regions, it's a beta-1 homolog, prevented binding. So this is the heel mutations where the heel residues were important, and that gave us a heel to toe interaction analogous to that of beta-1 to gamma-1. So now I want to turn to looking at a muscular dystrophy and the generation of what Karen has called Frankenstein proteins, which I sort of think of as proteins that maybe nature should have thought of making but never did. So the first one that was generated, which in our lab slang we called F1, was alpha-LN nitogen, and it consists of the LN and LEA domains of the alpha-1 chain of laminin fused to the G2 to G3 domain of nitogen-1. And this turned out to be very useful in further pursuing the issue of establishing the specificity of laminin polymerization LN interactions. However, it was too big for when we later on wanted to put it into AAV. Adenovirus has about a 4.8 kilobase total DNA limit in it. And when you try to exceed that, problems arise. So the basic model is this protein, or its shorter version, acts very much like a synthetic short arm that is bound through the nitrogen binding locus. And it will enable a non-polymerizing laminin with a defect in the alpha chain, whether it's alpha-1 or alpha-2, to then go ahead and polymerize. We evaluated this in vitro in dorsal root ganglia cultures. These were produced from the laminin gamma-1 flox mice, and then we would hit it with adenocree, and in order to get myelination, you have to provide the now missing laminin. And lo and behold, laminins 1 and 2 work very well. The red staining is the myelin. The green staining is the neurofilament staining to show the axonal structures. Well, first of all, if the laminin didn't polymerize, the Schwann cell precursors would not proliferate. And if a non-polymerizing laminin was used, no myelination occurred. This, however, could be restored with the alpha-LN nitogen protein. The correspondence of this in mice, and I believe also in humans, is that you see these amyelination plaques that develop, shown in the methylene blue section on the left and the EM on the right, where you see just these large groups of naked axons of various calibers. And they're not myelinated when they should be. So now I want to turn to the two alpha-2 laminin deficiencies. The first one is a model for the ambulatory dystrophy, which is milder. Its model, or a model for it, is the DY2J mouse, in which it was previously shown that it has an in-frame deletion in the alpha-2 LN, resulting in subsequent degeneration of the LN domain. And so it's a model for a dystrophy resulting what appears to be a purely polymerization defect. And in humans, there's a whole cluster of mutations, mostly missense mutations, in which one sees the human disease. So what we did is we inserted the shorter version of our alpha LN nitrogen, which I'm going to call F4, and injected it at postnatal day 1 into the dystrophic pups, basically. And then we followed the mice from about weaning on. And there were these dramatic changes. By immunofluorescence, we saw that it increased the alpha-2 levels. The alpha-2 levels, we still have a laminin alpha-2 present. It's just missing its LN domain. What was striking, though, especially were the peripheral nerve branches of the sciatic nerve, in which, without polymerization, the staining for laminin alpha-2 goes down practically to nothing, and it greatly increases with the treatment. Histology, so that's picroserous red, which stains the collagen. The muscle is enormously fibrotic, and this is prevented with the treatment. And the methylene blue staining shows that although you see these amyelination plaques in the untreated, they completely disappear in the mouse model. This is simply grip strength measurements. I'm showing it only for forelimb. The black is wild type. The blue is the untreated dystrophic. And depending on the concentration of virus, you would get great improvements in the grip strength. Whoa, what happened there? Well, despite that they look funny, one of the mice was not treated. The other two are DY2Js that were treated. And yes, there is a big difference, but they shouldn't look like that. Next, I want to turn then to the much more severe dystrophy, which has been called the non-ambulatory dystrophy because humans never gain the ability to leave a wheelchair. And this is a full-blown congenital muscular dystrophy. Kids get it at an early age, and they survive longer than they used to, but they generally die of respiratory failure. They have these terrible contractures, and like the ambulatory version, they have seizures. And here, the DY3K model is used, and that's the total knockout of the alpha-2 chain. And what happens is we see replacement of laminin alpha-2 by laminin alpha-4, which is normally present in the microvasculature. So we needed a different approach because of laminin alpha-4 attributes. It can't polymerize because it lacks an alpha short arm. It can't bind to dystroglycan. And in the 4-1 alpha, the laminin 411 version, it binds poorly to integrins, whereas the 421 version has okay interactions. And what in the mice, we saw that it greatly increased lifespan. Untreated DY3K are half dead by 4 weeks and don't live past 8. And here we had them going out for months if we use the double therapy, with the double therapy being alpha LN nitrogen and a protein called miniagrin that was developed by Marcus Ruge. It binds to the coil and binds to dystroglycan. The weights were improved, and a forelimb grip strength was improved. But this is not as good a fix as we saw with the DY2J mice. You have to remember, this is a structural repair. For a structural repair, you need enough protein to do the repair. And that creates an issue in terms of concerns about AAV toxicity. All right, so these are 3 sets of mice. There's one wild type in each, and the other one is dystrophic. Oh no. Okay. Even the ones that are treated only with F4 walk better than the untreated ones. All right. And this is my last slide. I think it's my last slide. It's simply showing sections of the peripheral nerve, sciatic nerve. And the point I wanted to make here is that the double treatment certainly eliminates the appearance of these naked axons. But for that matter, if you only treat with the F4 protein fixing polymerization, you also prevent that from developing. So here I want to thank the key person in my lab. She's been with me for 25 years. This is Karen McKee. And she has worked out many of the conditions to make these experiments work. And then I have my collaborators, Erhard Hohenester. That was is a lot of fun. Eric Kulsik for the cryo-EM, Marcus Ruegg, Judith Reinhardt for Miniagrin, and Raphael Reuten. And finally, my grant support. I had a 38-year-old run on the upper NIH grant, which just ended last year, and a new grant that has started this year, which is to focus on the dystrophy. And thank you for listening. Whoa. I didn't think it would go this late, the session.